New MCAT Cram Biology Flashcards Card 8 Polyspermy Prevention. All right. Identify the fast and slow process for polyspermy prevention. I'll give you a moment to recall the names of these processes. Okay, so first you have the acrosomal reaction, and that's the fast process. And then next you have the slow cortical reaction. So let's just delineate these two processes step by step, okay? So here you can see you have sperm binding receptors on the egg plasma membrane or embedded in the egg plasma membrane, okay, prior to fertilization. Then you have the approach of the sperm cell, and here you can see the acrosome in the tip of the head, hence the name acrosomal reaction. Okay, and the acrosome is an organelle that develops over the anterior half of the head of the spermatozoa, and its cap-like structure is derived from the Golgi apparatus of the cells. And here you can see in the next step, it's spilling hydrolytic enzymes into the jelly coat of the egg in order to penetrate it, okay? So this is what happens when it makes contact with the egg. And then what the what the spermatozoa does next is initiate the acrosomal reaction. So after a spilling of the hydrolytic enzymes into the jelly coat, or simultaneously rather, actin filaments from the acrosomal process start to grow and protrude, and this structure protrudes from the sperm head and penetrates the jelly coat, and it's going to bind to the sperm receptor in the egg cell membrane and extend through the vitelline layer. Okay, so it's beginning the process of fusion. The third step, which we can call contact infusion of sperm with the egg membranes, what happens is a hole is made in the vitelline layer. Okay, so this purple strip here is what's the vitelline layer. Okay, and what this does, it allows contact and fusion of the gamete plasma membranes. These membranes then become depolarized, and it's this quick depolarization that results in a fast block to prevent other sperm from fusing, okay? Because an electric potential is created that repels other inbound sperm, okay? So that's the prevention step that occurs as a result of the acrosomal reaction. And then you have next entry of the sperm nucleus, all right? And then last, you have the slow step or the cortical reaction. And this happens when it actually is initiated when the gamete plasma membranes fuse. It triggers an increase in calcium ion release in the egg cytosol. Okay, this is the cytosol, the liquid portion of the cytoplasm. And what this does is the calcium ion release then triggers a release of these granules, cortical granules in the egg. And what they do is they fuse with the plasma membrane and discharge their contents. And this leads to the swelling of the perivitelline space. Okay, and by swelling and hardening of the perivitelline space, this hardens the vitelline layer, and what it's going to do next is clip the sperm binding receptors, all right? And this is what results in the block of further fertilization of other sperm cells, hence the prevention or slow block of polyspermy. Because if there are no receptors for binding, then other inbound spermatozoa can't bind, okay? I know this was a little lengthy, but this is a step-by-step reaction that explains essentially how we came into being. Of course, if you have a twin or triplets or something, the process went a little haywire. There are several ways of getting duplicate babies, and when this goes wrong, it's one of the ways. I can't remember. It's like maternal twins. If your twin looks different from you, then this obviously didn't work. Okay? All right.